So I want to take a little bit of a second to go through some carbohydrate chemistry, very biologically related. I guess we talked about amino acids a couple of lectures ago. So now we're going to talk about acetals, ketals, maybe that's a comma, and carbohydrates. What a title. Okay, so we're going to have some complicated structures, so I figured I'd keep this lecture short as opposed to appending it on to the last lecture. So the first thing I want to mention is that monosaccharides, or single sugars, to keep things simple, exist as hemiacetals. Hemiacetals, which are half acetals. And half acetals are when we have well, first of all, our carbon has a hydrogen on one side, an R group on the other side. Then we're going to have an OR group, which I'll make the R group different, along with an OH group. We're halfway to the acetal or ketal. We have an OH and an OR group. Okay, so monosaccharides actually exist as hemiacetals in nature, which is pretty cool. So if you actually look at the structure of a if you take the chemical formula and you try to build a structure based on what you know about its chemical properties, you might land on something like this. I'm going to draw this very suggestively. That's not good. I'm going to start from my chair structure and work my way back. We want to draw these things from a chair structure. Sorry, it's just easier in a chair structure. There we go. All right, so chair structure with 4 equatorial groups, and heck, let's go with the beta version. We'll make it 5 equatorial groups. What I want to point out here, though, is that this carbon that I'm highlighting with a dot, that carbon is a hemiacetal, and only that carbon is a hemiacetal in this particular structure, and this is beta glucose, and it's the D enantiomer. Don't worry about that. Don't worry about the beta. It's glucose. Everything's equatorial, except that hemiacetal carbon doesn't have to be if it doesn't want to be. In this case, it is. Okay, so what I was alluding to was that it turns out that if you look at this structure, before you put it in water and allow it to equilibrate, you can consider an open form where you've got, okay, so I've got this here, I need to go here, here, there's an OH, there's an OH. I'm redrawing this chair structure. But I've got an OH and an aldehyde. So this is an aldehyde, and this is the open chain aldehyde form. So it's important to note, here's what the important takeaway is that if you have a carbohydrate, they start off as this open chain regular aldehyde, something we're kind of used to drawing, and in aqueous solutions, which, think about it, waters, the solvent of life, these sugars are going to cyclize to form hemiacetals. And then additionally, during polymerization, those hemiacetals turn into acetals. So we have an aldehyde. The aldehyde gives rise to a hemiacetal, which makes sense. It's not a ketal, it's an acetal in this case. Now, as I was alluding to, monosaccharides, these single sugars, polymerize to form polysaccharides when the sugars just polymerize to form polysaccharides, and these polysaccharides are connected by acetal linkages. Okay, so let's look at that. If we take our beta glucose and we react it with another beta glucose, so here we go, chair structure alert, everybody's having fun. So here's my beta glucose. And I mix that with another beta glucose. You get better at drawing these things after you take biochem, because you have to be better. And then if you talk to the biochemists, they're like, I thought the organic chemists were going to teach everybody the carbohydrate chemistry. Okay. Anyway, here's where we're going to assess our linkages. We're going to have we're going to combine this position with this position. Now, if you notice on the right-handed position, we have an acetal present at the carbon that's highlighted in green, but the carbon that's highlighted in green on the right-handed structure is just a normal alcohol, right? That carbon just has one OH attached to it. Okay, so we're going to do a polymerization. I believe we form lactose maybe when we do this, maybe not. I have to review my disaccharide structures. Okay, so we've got our equatorial OHs. One of them is an equatorial CH2 to an OH. Now what we're going to do is connect that O. We're just going to go straight up and bump into another chair structure, violating some of, you know, everybody's recommended bond angles and that sort of thing. So we have now this new linkage that I'm going to highlight in green, and that new linkage has with it now a ketal carbon. So we go from an acetal into a we go from a hemiacetal into an acetal linkage. Make sure I captured that. Because we still have a hydrogen that's shown as being an axial substituent, ultimately derived from an aldehyde. But we were halfway there when we started at the hemiacetal, and now we're at an acetal. Okay, and so this acetal, it turns out, is the stable linkage that forms all of the polysaccharides. If you think about it, like cell walls in plants are just polysaccharides, a bunch of pretty stable, really stable molecules held together by acetal linkages, which, you know, as I indicated, can be a reversible bond, but given all of the properties of carbohydrates, these linkages become pretty strong, and we have to have some pretty special techniques to get them separated and digested and all of that sort of thing. So takeaway message here, real life example of acetal and ketals. You want to be aware of it. This is really important for understanding carbohydrate chemistry. But otherwise, that'll do it for this kind of quick look in. In the next lectures, we'll look at like synthetic applications of acetals and ketals. So until then, I'll see you next time.